Hello. So we'll begin our discussion of micronutrients involved in pathways of energy metabolism with our discussion on thiamine. To start with thiamine, we'll begin with a little history. Thiamine was actually first discovered as an essential nutrient in the late 1800s in animals when chickens were fed what was called polished rice, which was actually refined rice with the outer bran layer removed. So when chickens ate this polished rice, they actually developed a neurologic problem, which we now call in humans beriberi. And we'll see in the deficiency symptoms and conditions of thiamine deficiency, there are several types of beriberi that we're going to be discussing at the end of this lecture presentation. Even though it was first discovered as an essential nutrient in the late 1800s in this way, the actual vitamin of thiamine, its structure was not isolated until the 1930s. So we'll move on next to the structure and the different forms of thiamine, and then to the food sources, its digestion, absorption, and metabolism. Here we see the structure of thiamine, this water-soluble B vitamin. We see the characteristics of this structure, including a pyrimidine ring. We see a connection which is called a methylene bridge, as well as a thiazyl group. The reactive carbon is highlighted in red, which may be involved particularly in phosphorylation or dephosphorylation reactions. When we discuss each of the micronutrients, particularly the vitamins, it will be important to be familiar with characteristics of their structure. So for each micronutrient, we will be reviewing significant and notable structures such as this. For exam purposes, it will not be necessary to draw out the structures. However, it will be important to be able to recognize a structure when provided. We'll now review the different forms of thiamine and their conversion. So we see in this reaction free thiamine, which is the form of thiamine that is absorbed in the level of the enterocyte. And from here we see the conversion of thiamine to thiamine diphosphate or thiamine pyrophosphate, abbreviated as TDP or TPP. We see the enzyme catalyzing this reaction is thiamine pyrophosphokinase, and we see an investment of ATP and a donation from ATP to phosphate groups to form this thiamine diphosphate. We're going to see when we talk about the functions and mechanisms of thiamine and different pathways of energy metabolism that the form that will be functional in these processes would be the TDP form. So once absorbed through the enterocyte and transported into circulation, we'll see that free thiamine is converted to TDP so it can have its effects in the body. There is conversion from TDP back to TMP, thiamine monophosphate. We see a phosphatase enzyme, thiamine diphosphatase, is involved with removing a phosphate group from TDP to form TMP. There will be some functions related to TMP, and also you may find TMP in circulation. So this is the enzyme dephosphorylating TDP to TMP. And then we see further dephosphorylation back to the free form of thiamine by the enzyme thiamine monophosphatase, so removal of an additional phosphate group to get back to the free form of thiamine. We see further phosphorylation of thiamine diphosphate or TDP to TTP thiamine triphosphate, with this reaction catalyzed by TDP ATP phosphoryltransferase. So again, we see an investment of an ATP donation of one phosphate group to the thiamine diphosphate to form thiamine triphosphate. There will be some unique functions that will require the thiamine to be in the triphosphate form with the 3 phosphate groups attached, and so we'll see that when we talk about the functions related to thiamine. We do see that there is process converting TTP back to thiamine diphosphate by this thiamine triphosphatase enzyme removing a phosphate group. It is interesting to see with each of these dephosphorylation reactions very specific phosphatase enzymes that will be catalyzing each. So we see the thiamine diphosphatase, thiamine monophosphatase, thiamine triphosphatase. We will now talk about food sources of thiamine. So this is where we're going to be finding thiamine at significant levels in the food consumed. So we can find thiamine in different meat products, particularly pork. Also, there'll be significant amounts of thiamine found in beans and legumes, as well as grains, whole grains, enriched or fortified grains. We'll see that the plant sources of thiamine will be in the non-phosphorylated form. So when we consume thiamine from plant sources, it will be as free thiamine. However, when we consume thiamine from animal sources, most will be consumed in the TDP form. And this makes sense because just as humans, we see that animals do have conversion within the body from free thiamine to TDP. This is done by the liver. And then the TDP we'll see is going to be active in the muscle and other tissues in the body. And when we consume muscle tissue from animal protein, we again will be consuming that in the TDP form. Since as mentioned previously, we absorb only free thiamine, since when we consume thiamine from animal sources, it's going to be in the form of TDP. This form of thiamine will need to be hydrolyzed, so broken down in the intestine to liberate the free thiamine from the TDP, so have to cleave the phosphate groups to prepare for absorption. We'll talk now about the process of absorption. We see that thiamine is absorbed through the jejunum, involving carriers THTR1 and THTR2. This is an active transport process again through these carriers THTR1 and THTR2 found in the brush border membrane of the small intestine. THTR1 is also embedded within the basolateral membrane of the small intestine, which then allows for transport from the enterocyte into circulation. While much of the thiamine that we consume is absorbed, so the percent absorption is fairly high, there are some what are called anti-thiamine factors that reduce its bioavailability. So we see what are called thiaminases, which are enzymes found in food that actually cleave thiamine, destroying the compound, and this reduces absorption. We also see what is called the polyhydroxyphenols, also reducing bioavailability of thiamine. We see that thiaminases found in raw fish catalyze the cleavage of thiamine, destroying it. However, cooking the fish deactivates the enzyme. Polyhydroxyphenols are found in coffee, tea, nuts, and some berries, and inactivate thiamine by destroying the thiazyl ring structure. Additionally, absorption can be reduced by excessive intake of alcohol. This particularly affects movement of thiamine from the enterocyte through the basolateral membrane and into circulation. Now we'll talk about the transport and storage of thiamine. It's transported through circulation as free thiamine, thiamine bound to albumin, or as thiamine monophosphate, so attached to one phosphate group. Once absorbed, most free thiamine is taken up by the liver and phosphorylated to the functional form involved in pathways of energy metabolism, TDP, and this is catalyzed by pyrophosphokinase. We see that thiamine is stored in the liver, the skeletal muscle, and the brain. So now we'll discuss the functions of thiamine in the body, particularly in pathways of energy metabolism. We'll see that thiamine is primarily involved in decarboxylation reactions, so removing carbon dioxide. This is going to be present particularly in dehydrogenase complexes. So we see here the pathways of glycolysis. We see Krebs cycle. We also see the hexamonophosphate shunt. Areas highlighted in red is where we will see involvement of thiamine, particularly in the form of TDP. So we'll first look at the two decarboxylation reactions in the dehydrogenase complexes. So we see the pyruvate dehydrogenase complex. We also see the alpha-ketoglutarate dehydrogenase complex. In each of these reactions, we see removal of a carbon in the form of carbon dioxide. So again, this would be called a decarboxylation reaction. And TDP, so thiamine in the form of thiamine diphosphate, is involved as a cofactor, assisting the enzyme catalyzing the reaction. And so we see this similarity in terms of the removal of carbon dioxide removing carbon. So we see here pyruvate is a 3-carbon compound going to acetyl-CoA, which is a 2-carbon compound. We'll see a similarity with the alpha-ketoglutarate dehydrogenase complex. So we see that alpha-ketoglutarate is a 5-carbon compound, and when we remove a carbon in the form of carbon dioxide, it's converted to the 4-carbon compound succinyl-CoA. So just like in the pyruvate dehydrogenase complex, in this alpha-ketoglutarate dehydrogenase complex, thiamine in the form of TDP is acting as a coenzyme to the enzyme catalyzing the reaction alpha-ketoglutarate dehydrogenase. Additionally, we see involvement of thiamine in the hexomonophosphate shunt, where we see formation of NADPH. The enzyme catalyzing this reaction is transketolase, and transketolase is thiamine dependent. So here we see further description of thiamine and its coenzyme role and its involvement in transketolase, which again is the thiamine dependent enzyme. And this is going to have the implications of pentose and NADPH synthesis in the hexomonophosphate shunt. We also see non-coenzyme roles of thiamine, particularly in membrane and nerve conduction, and this will be in the form of TTP rather than TDP. So in this form of TTP, we see that thiamine is involved in activating ion transport and ion transport related to nerve conduction. In neurons particularly, it's thought to activate chloride transport. We see that also in the form of TDP involvement in the regulation of sodium channels and acetylcholine receptors for nerve impulse transmission. We'll now go through the recommended dietary allowance, or RDA, for thiamine. You see that for men, the RDA is 1.2 milligrams, for women, 1.1 milligrams. Pregnancy, it's slightly higher, 1.4 milligrams, and also lactation, it's slightly higher, with the RDA being 1.5 milligrams. There's little danger of toxicity for thiamine, and there is no tolerable upper intake level for thiamine. We now see deficiency symptoms of thiamine. So beriberi is going to be the key and primary clinical condition associated with thiamine deficiency, and we'll discuss 3 beriberi. The first is called dry beriberi. We'll also discuss wet beriberi, acute beriberi, as well as the Wernicke-Korsakoff syndrome. Dry beriberi is associated with chronic low intake of thiamine, and this is particularly seen in older adults. Clinical symptoms of this would be muscle weakness, muscle wasting, and peripheral neuropathy. We see further cardiovascular system involvement in wet beriberi. So in addition to the peripheral neuropathy, that would be similar to the symptoms of dry beriberi, we also see involvement of the heart with a symptom of cardiomegaly, or enlarged heart, and tachycardia, which would be a rapid heartbeat. In severe cases, this could also lead to heart failure. Another subtype of beriberi is called acute beriberi. This is seen mostly in infants. Symptoms would be anorexia, so lack of hunger, vomiting, and lactic acidosis. And this is attributed primarily to parenteral nutrition solutions low in thiamine. Lastly, we'll see the Wernicke-Korsakoff syndrome associated with alcoholism, so chronic high intakes of alcohol. We see the symptoms being paralysis of ocular muscles, involuntary eye movement, and impaired muscle coordination. We also see severe neurophysiological complications.